Hi everyone. In this presentation, we're gonna talk about OpenHI, histopathological image platform for digital pathology. My name is Pagon Puttapirat, and our team is at Sian Jiaotong University. Before we go to OpenHI, I'd like to talk about whole slide image, or WSI. This is different from radiology scans. It is originated from the glass slide, and it is scanned by a whole slide image scanner. This results in a really large 2D digital image. Before the development of OpenHI, we have two existing software. First is QPath, and the second one is Cytomine. QPath is a single machine software, so when you install it on your machine, it will run locally. It has a lot of useful functions like annotation and analysis. However, it is not so easy if we use it collaboratively. For Cytomine, it is closer to OpenHI because it's a cloud-based software and it has different modules to do different things. But there are differences, and we'll discuss these unique features of OpenHI in this presentation. Before we go to that, I would like to talk about how we think the digital pathology platform should work. It should consist of three things: scanning, management, annotation, and analysis. In scanning, obviously that would consist of whole slide image scanner and its software to control the scanner. Ideally, in management, we hope that the DICOM server would dominate this area. And finally, in the annotation and analysis, right now you can use QPath to do a lot of that. You can also use Cytomine in between here, management and analysis, since it has different modules to deal with different tasks. The problem is none of the components in each area really works with each other. For example, there are 8 or more whole slide image scanner vendor, and most of them doesn't work with each other, and I think we to our knowledge, none of them works with the DICOM server. And you cannot really retrieve image from the DICOM server to QPath. So we hope to develop an interconnected environment for the digital pathology platform. So here's our view. We see OpenHI as the whole slide image client, and it would connect to everything using a standardized protocol and file format like DICOM and PACS. OpenHI could reside in the server as the DICOM server extension to help additional processing and analysis. So here's the underlying architecture of OpenHI. It is written mainly with Flask, which is a Python-based web framework, and it results in two main things. First is the GUI for annotation and analysis, and the REST API that other applications could interface with OpenHI. So here are some unique features. First, I would like to talk about resolving power-based virtual magnification. Why do we need virtual magnification? Because some pathology guidelines require pathologists to rely on optical magnification to make the diagnosis. So in this method, we think of a single resolve unit of light. Right here, we take each pixel and present it on the screen. When we present it on the screen, we can trace back to where each pixel came from. We don't only care about the scale, we care about the resolving power and the single unit of the pixel which is on the screen. With this method, we have found out that only 20 to 40% of the diagnostic area viewed on the screen could is from the optical image. So another unique feature is about macro and micro annotation. In a given whole slide image, we can do the annotation at the pixel level, at a cellular level, right? And when we group a lot of cells together, we get a diagnostic area. And in one slide, we have a lot of diagnostic area. You can imagine for data scientists that if this is a whole slide image and you do an annotation on interested regions, then the rest would be unknown. In OpenHI, we have whole slide image. We scope them as a diagnostic area first, then we do annotation. The rest would be the unannotated part, and the rest outside DA would be the unknown area. So as a result, we hope that this system would help pathologists cope with their daily working routine and make better diagnosis. It should be easily deployed on a medical institutes, since it requires only standard Linux environment. For data scientists, we hope to create a complete annotation and analysis environment for everyone. In conclusion, we hope that better standardization and machine learning methods combined with conventional pathology would be transformed with OpenHI to digital pathology, and we could have better tools for sharing knowledge and open and standardized analysis. To end, you could see source code and documentation at aipath.org/OpenHI for more information. Thank you.